A very good evening to everyone. Welcome to this NPTEL Week 1 live TA session for the course Cell Culture Technologies taught by Professor Moinak Das from IIT Kanpur. My name is Ankita De and I'm a Prime Minister's Research Fellow pursuing PhD at the School of Medical Science and Technology, IIT Kharagpur, and I'm going to be one of the TAs for this course this semester. So I'll be hosting this live TA session every week on Tuesdays 6 to 7 p.m. So let's have a quick look at the session outline. So this is going to be a live interactive 1 hour video session during which we will be solving some previous year's assignment questions from the year 2021 live. That will in turn help you in solving some of this year's assignment questions, the weekly assignment questions. For any doubts or clarifications regarding the solutions, you may use the chat box option to post your queries and I shall try to answer them. Towards the end of the session, we'll also have a Q&A segment where you can post your queries and I'll try to address them. In the end, we will provide you with the necessary course materials and class recordings for your access later and the link to the YouTube link to the video which is going to be recorded. So let's get started. So before we move on, before discussing the assignment questions, let's just quickly recapitulate few key points that were discussed in this week's lectures. So some of the key points that were discussed in this week 1 lectures. First of all, we learned about what is cell culture and its various applications. So cell culture is basically it refers to the laboratory methods that enable the growth of eukaryotic and prokaryotic cells in an artificial environment outside the body under physiological conditions. It has several applications, such as it is used as a model system to study the basic biology of cells or to replicate and understand certain disease mechanism or for the development of vaccines or as a bioreactor for the production of large scale for the production of biopharmaceuticals in a large scale or to determine the toxicity of several novel drug compounds. So we also discussed few terminologies in with respect to this cell culture, such as the primary versus cell line versus cell strain culture, and some of you had certain doubts regarding this. So we'll try to go over each one of them and try to understand what each one of them means. So we have discussed about what primary cell culture is. So primary cell culture, we'll try to understand this with the help of this model. So when we are taking cells directly or we are isolating cells directly from out of an organ, and we are trying to culture it in a plate. So when we take a portion of a tissue out of an organ and we disaggregate the cells and make it into a single cell suspension and we culture it in an artificial environment outside the body in a culture plate. So this is called, this stage of cell culture is called primary culture, when you are taking it directly from out of an organ. So this is referred to as primary culture. So these cells are cultured on the surface of a culture plate. So gradually the cells will divide and it will cover the entire surface of this culture plate. Now once the cells have covered the entire surface, we need to subculture the cells. This process is also known as passaging or splitting. So by splitting or passaging or subculturing, we mean that when the cells on the surface of a culture plate has fully occupied the entire available surface, we need to split it into further secondary culture, because cells, after they have reached a certain stage, that is they have entirely filled up the surface, they cannot grow further, because there's this phenomenon that occurs, that is known as contact inhibition. So under that condition, cells cannot grow further and their rate of growth decreases. So in that case, we need to subculture the cells. So after you have subculture the cells, this primary culture becomes your cell line. Okay, so but this cell line you can go on culturing like this, like from one culture plate we are subculturing or splitting the cells into four culture plate, and eventually these four culture plate we can split it into 16 subculture plates, and so on. So as the cells keep on dividing and keep on covering the entire surface of the cell culture plate, we need to subculture the cells, because the cells will only proliferate properly when they are in the log phase of their growth, that is when they're in the exponential phase of their growth. So there is also this term we are discussing in with relation to this, that is the confluent culture, that is when they have covered the 100% area that is available to them in the culture plate. Now cells need to be cultured when they have almost reached about 100% confluency. So suppose we say that the cells are 90% confluent. By this we mean that it has covered only 90% of its available surface area. So this is how it happens. Now, say you want to study the expression of a certain gene of a certain gene, and you want to understand its mechanism in what role it plays in a particular disease. So to understand the role of that gene, what you need to do is, either if that gene plays some essential function, you can knock down the expression of such a gene by introducing some foreign material into the cell which will knock down its expression. So how are we going to do that? So to do that, what we do is we take there are certain molecules that are available. I won't go too deep into that. So these are known as the siRNA, that is these silence the expression of a certain gene. So we inject these foreign particles inside the cell, and we turn off the expression of a certain gene, and we see if the particular phenotype, that is the particular behavior of the cell, is changed or not. So that is how we study the expression of a gene. Now suppose we have a pool of cells, and from amongst these pool of cells, you suppose we are injecting a particular this particular silencing RNA, that siRNA, into these cells, and we want to study we want to turn down the expression of that gene. So when the cells will not easily accept this foreign material inside of it, so it will try to reject that. So when we are trying to do this, majority of the cells won't take up this foreign material. So say only out of 100 cells, only 10 cells will take up this foreign material and show silencing in gene expression. So from amongst this pool of cells, you need to select for those 10 cells. So this selection procedure is known as positive selection. Like, you will positively select for those cells which has taken up this foreign material and has successfully has shown a turn off in its gene expression level. So this is referred to as a cell strain. That is that subpopulation of cells which you have positively selected from out of that pool of cells, and therefore this is referred to as a cell strain. Now we will come to the other terminologies, that is finite versus continuous cell culture. So when you are isolating the cells from a healthy organ or normal organ, and the uh, cells are growing in culture, the cells will grow only for a finite number of times. That is, they will have a limited lifespan. But however, if you isolate the cells from a organ having a tumor, they will they will show this property, that is they can be grown indefinitely in culture. That is, they can be grown for a number of times in culture, and these cells are said to have this cell line is said to be a continuous cell line, because you can go on proliferating them, you go on dividing them, culturing them in the culture plates. So this is the difference from a finite versus the continuous cell line. So now we'll come to the other terminologies that were discussed. So we have also discussed about the culture conditions. Culture conditions are the necessary nutrients which are required for the cells to grow outside the body. Just as the cells inside our body require nutrition to grow, similarly, when you are culturing the cells outside of our body, they will also require certain nutrients to grow, such as amino acids, vitamins, and enzymes, hormones, oxygen, carbon dioxide. So these are the substances that we need to supply in our cell culture plate in order for the cells to grow. Now we have also come across this term, that is cryopreservation. Some of you also have had a confusion regarding this term in the discussion forum. So first and foremost, we need to discuss what is cryopreservation, why we need to do it. So as we know that cells cannot be kept in culture continuously if not needed, because that would be a waste of both time and resources. So cryopreservation is the method that comes into play here. It is the technique by which cells can be preserved for long-term storage at sub-zero temperature, usually below minus 130 degrees centigrade. So we require appropriate freezing medium for that, and for this purpose, which will lower the freezing point of the medium and will slower the rate of cooling to prevent the crystallization of ice in the within the cells. So cryopreservation is done so that whenever you have a surplus amount of cells, you can store it, and then later when you again require it, you can take it out of that minus 130 degrees centigrade and you can culture it. So that is the purpose of cryopreservation. So someone is asking, is session 1 and 2 the same? No, they are not the same. Session 1 is taken by another T and session 2 is taken by me. So you can join either of them. So next we will discuss about this. We have discussed about the cell cycle concept and the different phases of the cell cycle. So basically the cell cycle is divided into two phases, the interphase and the mitotic phase. So during the interphase, which was earlier known as the resting phase, but although it might apparently look like it's resting, but it is not. The interphase is a very active stage, because during this phase, the cell prepares itself for the next division cycle, and then we have the mitotic phase or the M phase. So during the M phase, the cells actually divide. So there are these phases in turn are divided into several sub phases. Like the interphase is divided into the G1 phase, the S phase, and the G2 phase. So, and the M phase is further divided into four phases: the prophase, the metaphase, the anaphase, and the telophase on the cytokinesis. So before a cell enters the cell division cycle, that is the mitotic phase or the M phase, it prepares itself for division. How? It replicates its DNA, which occurs primarily in the S phase of the interphase, and also duplicates its centriole. And after it enters the M phase, the chromosomes will arrange themselves on the equatorial plate, and they will be pulled towards the opposite pole, and then they will be divided into two daughter cells. Now, whether a cell at this point, whether that is at this point where a cell is entering the interphase after having undergone one cycle of division, it is offered two options. That is, it can re-enter another cycle of division, or it can opt out of this cell division process. Now, what are the factors which determine whether the cell will opt out of division or it will re-enter another cycle of division? Now, there are several checkpoints at the beginning of each of these phases, like at the beginning of the S phase, at the beginning of the G1 phase, at the beginning of the M phase, which determine whether the cell shall enter the next phase or not. Now, only if the cell meets certain requirements, only then it can move on to the next phase. So, this is determined by the cell cycle checkpoints. Now, at the beginning of any division, that is after the cell has undergone one cycle of division and it enters the interphase, it can either, as I said, opt out of division and enter a G0 phase, which is also known as a quiescent stage. That is, it can temporarily uh, opt out of the cell division cycle and enter an inactive state where it does not divide. Or it can permanently opt out of division, that is, it will never re-enter the cell cycle again, or it can enter the cell cycle. So there are these two options, that is either it can opt out of cell division either temporarily or permanently, or it can re-enter the cell cycle. Now, what are the factors which determine this? So, so before discussing this, we also need to understand that when does a cell decide to divide and when does it decide to not divide. So here we introduced you to a few other terms, that is differentiation, de-differentiation and potency. So now we have introduced you to some more terms, that is potency, differentiation and de-differentiation. So differentiation is basically the acquisition of more specialized characters of a progenitor cell. That is, say at the beginning of when in the zygote stage, that is the zygote which is formed by the union of the male and the female gametes. So at that time, a cell is said to be the most potent. That is, potency refers to the ability of a cell to give rise to all other kinds of cells. So the zygote has the maximum potency, and so it is said to be totipotent. That is, it can give rise to almost all other cells in the body. So gradually, this potency or the ability of the zygotic cell decreases. That is, as the zygote begins to divide and it divides to give rise to 2 cells, and then it will go on to give rise to 4 cells, 16 cells, like that. It potency, that is this ability to give rise to other kinds of cells in the body, will also decrease. So the zygote, when it has reached this stage, that is the blastula stage, it has it is primarily made up of two parts, that is the inner cell mass, and then we have the outer cell mass. So this inner cell mass is composed of the embryonic stem cells, and the outer cell mass will give rise to the trophoblast, which will give rise to the extra embryonic tissue. So these pluripotent cells are these embryonic stem cells which are present inside this which are arise from these inner cell mass, are pluripotent in nature. That is, they can give rise to some but not all kinds of cells in the body. Like the pluripotent cells is capable of forming the embryo proper, which later gets implanted into the endometrial lining of the uterus. However, it cannot give rise to the extra embryonic tissue. Therefore, it is called pluripotent. Thereafter, as the potency of these cells further go on decreasing, it will become multipotent. That is, it's the multipotent cells. An example of the multipotent cell would be the hematopoietic stem cell, which can further give rise to all kinds of blood cells in the body. The multipotent cells will only be able to form the spells of a particular lineage. Okay, so so as you see that the potency of a cells will go on decreasing, that is, its ability to give rise to other kinds of cells will go on decreasing. And then at the last we have the unipotent cells. Now unipotent cells are capable of self renewal, that is, they can give rise to cells other cells of their own kind, and they can differentiate to form only one kind of cell. So that is why they are being termed unipotent cell, that is they can give rise to only one kind of cells. So these stem cells, as they go on proliferating, their potency shall decrease, and by decreasing we mean as the cells will go on acquiring more and more specialized characters, it is that is it becomes more and more differentiated. So differentiation is basically, as I have already mentioned, the acquisition of more specialized characters. So as the cells become more specialized or differentiated, their potency shall decrease. And sometimes when a differentiated cell, that is when a cell which has acquired the highest level of specialization is it can be made to de-differentiate, that is it can be induced to acquire a more progenitor state, and it could be induced to induced to acquire a more primitive state. So this stage is known as the this process is known as de-differentiation. So as I was mentioning here, that what are the factors which determine whether the cell will re-enter division or it will exit the cell cycle. So whether this cell will re-enter division is determined by whether the cell is terminally differentiated or not. That is, whether it has reached the highest level of specialization or not. So if it has already reached the highest level of specialization, then it won't divide further and it will exit the cell cycle. So examples of those kinds of cells are the, say, the neurons, which do not divide further. So they are in a permanently non-dividing cells. So these are the cells that we call the non-dividing cells. And the dividing cells are those which have not yet reached the process of, which have not yet reached the phase of differentiation. That is, they are in a more progenitor state and they can go on dividing to give rise to more cells of their own kind, or they can differentiate to form a more specialized kind of cell. So these are the terms. Now we have also come across this term, that is adherent versus non-adherent cell. So adherent cells are those which require a surface area to grow. That is, they require a substratum to grow. As I've already shown you in this picture, that the cells grow on the surface of a culture plate. So these cells which require a surface on which they can adhere and grow are known as the adherent cells, whereas cells which do not require a surface to grow are known as the non-adherent cell or the suspension cells. So now we will quickly move on to discussing the previous year's assignment questions. So the first question is, which of the following is not a phase of the cell cycle? And we have four available options here: interphase, mitosis, gap zero phase, or the open phase. Now you can try to answer this question by typing in the chat box like which one you feel is the correct option, and then we shall discuss. Okay, so yeah, so I do see a couple of answers in the chat box. So you are saying it's going to be open phase. So we'll discuss why. So as we have already discussed in one of the previous slides, that interphase is the phase during which the cell prepares itself for the next division cycle. So of course it is a phase of the cell cycle. So this is not the correct option. The mitosis or the M phase has again another phase of the cell cycle or the dividing phase. The gap zero phase, as we have discussed, is the inactive stage. That is when the cell chooses to opt out of the cell cycle and enter an inactive state. It is known as the gap zero phase. So uh, I need to mention this now. That is, this gap zero phase can be either reversible or it could be irreversible. That is, the cell could be in a temporarily gap 0 phase, that is it could be temporary G0 phase, it can later re-enter the cell cycle division process, or it could be in a permanent G0 phase. So this G0 phase could be either reversible or irreversible. So the only option that we are left with is the open phase. So open phase is the right option. So now we'll move on to the next question. DNA replication occurs under which phase of the cell cycle? Is it the gap 0 or the G0 phase, the gap 1 or the G1 phase, the synthesis or S phase, or the mitosis or the M phase? So again you can answer. Yes. So I do see a number of right answers in the chat box. So yes, it is the synthesis phase or the S phase. So we have already discussed in the previous question and in the previous slides that how the G0 phase and the G1 phases, about the G0, G1 phases and the M phase. So S phase is the phase where DNA synthesis or DNA replication takes place. So DNA replication occurs under which phase of the cell cycle? It is the synthesis phase or the S phase. Okay, now we'll move on to the next question. On dedifferentiation, the potency of cells, and then we have a blank. So now we have four options here: increases, decreases, does not change, or none of the above. So again, you can answer in the chat box. That is, on dedifferentiation, the potency of cells shall increase or decrease, or it does not change, or none of the above. Yes. So again, I see a number of right answers in the chat box. On dedifferentiation, the potency of cells increases. So this I will explain again with the help of the previous slide. That is, as I've already explained, that during differentiation, the cells become more specialized. That is, they acquire more specialized characters from a low level of specialization. They acquire a more specialized level of phenotype. So dedifferentiation is the exact opposite of differentiation, and when we say that a cell is dedifferentiating, we say that it is acquiring a more progenitor state, or its potency increases. So it is going upward. So as it goes upwards, its potency shall increase. Okay. So here I have provided you with a note which you can refer to later. So this discusses the different types of potency and the different types of cells. That is the totipotent, pluripotent, and the multipotent cells with along with the example. So you can refer to this later. Now we'll move on to the next question. On differentiation, the function of cells becomes less specialized, more specialized, unknown, or has no correlation to specialization. So you can answer now. It's also somewhat like the previous question. Yes. So on differentiation, the function of cells becomes more specialized. True. So we have already discussed this in some of the in our previous question and the previous slide. So now we'll move on to the next question. Which of the following is a marker of induced pluripotency? SOX9, KLF8, OCT4, or MHC? So you have four options here. Now, before discussing this question, I want to discuss another topic, which is about the induced pluripotent stem cells, which I had left out in my previous discussion. So before discussing this question, let's quickly understand what induced pluripotency is and what induced pluripotent cells mean. Yes. So as we have seen that we have discussed what pluripotent cells are. So the inner cell mass of the blastocyst can give rise to the embryo proper, as we have already discussed, and the outer cell mass gives rise to the extra embryonic tissue. Now these embryonic stem cells are said to be pluripotent. So later, a few decades, like in few years ago, in the year 2006, it was discovered that any cell in the body, any cell such as any somatic cell or any cell that you take out of your body, that which are the specialized cells, say you take out a skin cell or some other cell, which is a specialized cell, that is which is fully differentiated and has somewhat lost this ability, that is somewhat lost its pluripotent activity. So if you take that kind of a cell and you inject certain critical factors, so there are 4 critical factors. So if you inject these 4 critical factors into any somatic cell of a human, or say out in a mouse, so these cells were able to convert to a more pluripotent state. So since this pluripotency is induced, we call them the induced pluripotent stem cells. So, induced pluripotent stem cells, this technology was first discovered by Shinya Yamanaka in the year 2006. He's a Japanese scientist, and he was also offered the 2012 Nobel Prize for this technology, that is the induced pluripotent stem cell technology. So nowadays, induced pluripotent stem cells find their applications in a number of a number of applications, like organ tissue, organ regeneration, or tissue repair. However, we need to conduct more research in this regard before its true potential is realized. So now we will move on to the question that we were discussing. Sorry. Yeah. So these are the 4 factors. So which of the following is a marker of the induced pluripotency? So as I discussed that there are 4 critical factors which are required to induce pluripotency in any somatic cell. So what are these 4 critical factors? They are SOX2, OCT304, CMYC, and KLF4. So which one do you think is the correct option here? So you can write in the chat box again, and we'll move on to the discussion. Okay, so a couple of you are saying OCT4 is the right answer. So let's see. Yes, so as I've already discussed, OCT4 is the right answer. So as you can see in the Joyner figure, uh, when you are taking the cell, like any somatic cell out of a donor, and we take it out of its body, that is these are the somatic cells, and we inject these 4 critical factors or genes, SOX2, OCT304, KLF4, and CMYC, and these 4 critical factors are able to reprogram these somatic cells to a induced pluripotent phenotype. So thereafter we call these cells as the induced pluripotent stem cells. Now these induced pluripotent stem cells can be made to differentiate into many other kinds of cells in our body. It could be therefore used for, as I have mentioned already, organ or tissue repair, regeneration, and therefore this process is known as as we call it in modern research, regenerative medicine. That is, we are able to regenerate the injured tissues or organs from these induced pluripotent stem cells. So a couple of you have already discussed, as you have mentioned in the discussion forum, that about regenerative medicine. So this is what it implies: regenerative medicine with the help of induced pluripotent stem cells. And the factors, these essential genes or factors that are required for this purpose are known as the Yamanaka factors, after the scientist who first discovered them, that is Shinya Yamanaka. So now we'll move on to the next question. So GDNF is a growth factor that stands for good dermal nerve factor, glial cell line derived neurotrophic factor, glioblastoma dimensional notch factor, or glomerulus driven nephron factor. So you have four options here. It's a very simple plain question. You just need to tell what the full form for this GDNF is. So you can answer in the chat box and we'll see what the right option is. So as we have discussed previously, growth factors are essential for the for cell culture. So we need to supply the medium in which we are culturing our cells in outside the body with certain growth factors for their proper growth. So GDNF is also one kind of such growth factors that we require. Okay, so we will see what the right answer is. So GDNF refers to glial cell line derived neurotrophic factor. So it was first purified in the year 1993 from a glial cell line culture which promoted neuronal survival. So this factor is essential for the survival of the neurons. It can support embryonic midbrain, dopaminergic, sympathetic and spinal motor neurons and is required for the development of the enteric nervous system, ureters and kidneys. So GDNS refers to this glial cell line derived neurotrophic factor and it is essential for the growth of neurons and their survival. So now we'll move on to the next question. Question number 7. Actin is a sugar, lipid, carbohydrate or protein? So you can type in the chat box the right answer. What is it? And we'll then see what actin is and we'll discuss about it. Okay, so you are saying that actin is a protein. Now let's see what the right answer is. Yes, so actin is a protein. Now we'll move on to the next question and then we'll discuss about the importance of actin. So which of the following proteins is responsible for force generation in skeletal muscles? Keratin, myosin, collagen or insulin? So we have four options here. You can type the answer and we can discuss the correct option. So which of the following proteins is responsible for force generation in skeletal muscle? Okay, so we'll see what the right option is. So the right option is myosin, as some of you have already mentioned in the chat box. So myosin is the prototype of a molecular motor. That is, it is a protein which converts chemical energy in the form of ATP to mechanical energy, thus generating force and movement. So as you are aware, as we have discussed previously that about some of the non-dividing cells, while we were discussing the non-dividing cells, we have discussed about the neurons. So the muscle cells or the skeletal cells are also another form of non-dividing cells or a differentiated cell that has reached its highest level of differentiation or specialization. So how are muscle cells formed? So several muscle cells will aggregate to give rise to muscle fibers. These muscle fibers have a multinucleated appearance because they are formed by the union of several muscle cells. Now, each of these muscle fiber, which also means that, so here we need to understand that muscle cell refers to muscle fibers because muscle fibers are formed by the union of several muscle cells. So, muscle fibers are nothing but large muscle cells which are formed by the aggregation of several small muscle cells during the developmental phase. So they have a multinucleated appearance, and these muscle fibers contain are composed of bundles of single as a so these muscle fibers contain many myofibrils which are bundles of actin and myosin filaments which are organized into a chain of repeating units called sarcomeres. So these actin and myosin are mainly responsible for producing force during for generating force during a muscular contraction. So actin and myosin are the two essential proteins which are present, which make the myofibrils, which are in turn responsible for skeletal movement. So now we'll move on to the next question. So question number 9. In heart, cardiac cells contract in unison by followed by a blank through gap junctions. So we have four options here: cell valve current transfer, cell membrane current blockage, cell current transfer, or cell valve current blockage. So here we are discussing about the heart and the cardiac cells. So muscle cells, as we have discussed previously in the previous question, that the muscle cells can be the skeletal muscle cells. We have They are the voluntary, as you might have studied about this previously. So they can be broadly divided into three types. That is the skeletal muscle, which are the voluntary muscle, and they are also the striated muscles. That is, they bear certain cross striations, cross striated appearance. So they are also referred to as the striated muscles. So they are under our voluntary control. So they are also referred to as the voluntary muscle. And then we have the involuntary muscles. So they are the smooth muscles. So they do not have a striated appearance, and therefore they are called smooth muscle. So, and then we have the cardiac muscles. They also have a striated appearance. However, they are also not under our active control. So these are the three types of cells. So example would be of the skeletal examples of the skeletal muscle cells would be, as I've already discussed, the skeletal muscles. And the examples of the smooth muscles are the muscles which line the our gastrointestinal tract. So these are not under our control, and so they are referred to as the involuntary muscle. And then we have the cardiac muscle cells. So these are also another kind of differentiated cells, and so we will discuss about this in our discussion section. So now you can write the answer in the chat box and we'll see what the right answer is. So one of you has written that the right answer is cell current transfer. So let's see what the right answer is. Yes, the right answer is cell current transfer. So as I was discussing that the cardiac muscle cells. So the cardiac muscle cells are also referred to as the cardio myocytes. They are arranged in somewhat in this form, as you can see in this adjoining figure. And between these adjacent cardiac muscle cells or the cardiomyocytes, there are certain channels. So these channels are hexameric channels channels, and they are referred to as the gap junctions. So what is the function of these gap junctions? Now the gap junctions play a very important role. That is, as you know, it is the function of the heart is to pump blood throughout the body. So what these gap junctions do is that they enable the quick movement of small ions through the adjacent cardiac muscle cells, and therefore they facilitate in the conduction of electric impulse to the heart. So that is the purpose of the gap junctions. So where are these gap junctions present? So gap junctions are present in the region of the intercalated discs. And as it is given in the description here, the intercalated disc connects the cardiac muscle cell and consists of desmosomes and gap junctions. So gap junctions are nothing but they are channels to adjacent cardiac muscle cells, which enable the movement of small ions and molecules through the adjacent cardiac muscle cells and help in the propagation of electric impulse through the heart. So now we will move on to the last question. So what are cardiac cells called? So as I have already discussed in the previous question, so we have four options here: cardiomonocyte, cardiocells, cardioclusters, or cardiomyocytes. Let's see what the right option is. You can type in the chat box and we'll see. Okay, so the right option is cardiomyocytes. So as one of you have written in the chat box, so that is the right answer, and I've already discussed this in my previous slide. That the cardiac muscle cells are also referred to as the cardiomyocytes. So that was all of the questions from the previous year's assignment. So now we will have the quick Q&A segment where you can ask me your queries by posting them in the chat box, and I will try to answer them or solve them if possible, like if I will try to solve them. So before that, I would like to acknowledge NPTEL and IIT Madras for giving me this TA-ship opportunity. And I would also like to thank Professor Moinak Das, the course instructor for this course, for selecting me for this role. I would like to express my gratitude to my supervisor, Professor Mahitosh Mandal sir. I would also like to thank MHRD, Government of India, for the Prime Minister's Research Fellowship. And I would like to thank IIT Kharagpur for allowing me to pursue PhD at the institution. Lastly, I thank all participants for joining the session. So if you have any questions related to the questions that we discussed, you can post in the chat box and I will try to address them. Okay, so yes, some of you are asking about the link for the exam portal. So regarding the links and the exam dates or the last date of submission, you can find all of that in the announcement section of your course portal. That is, you can find them that once you log in into your NPTEL portal, you can go to your course, and there you will find several tabs. Over there, you can find the course announcement section, and there you can find all of them, like the link to the exam portal or the last date of registering for the exam and all those information. Okay, so I don't see any questions in the chat box, so we'll now end the session. So thank you very much all for joining, and I look forward to your participation in the coming weeks as well. So remember that this TA session will be taken by me every Tuesdays from 6 to 7 p.m. and you can join the session with the same link that you have used today. So see you again next week. Thank you.